Welcome back. In part 2 on our section of cells, we are going to focus on what you need to know about the microscope that has allowed us to understand about these building blocks of life, the cells. Okay, and just a reminder that a summary of all of this is available to save or print. Right, so the first thing is, can you label a microscope? We got the eyepiece at the top, which magnifies whatever we're looking at by 10. We have the coarse and fine focusing knobs. We have the arm that we use to carry the microscope with. We have the nose piece with the turret and the objective lenses. Now, if you remember, you always start with the smallest objective lens, which is 4 times, and then you go to the 10 times, and then you go to 40, each time making sure your focus is absolutely beautiful. You put the microscopic slide with the specimen and the cover slip, which usually has some dye on it to highlight certain parts of cells. And you put this onto the stage with a little clip to keep it in place. There's a diaphragm, just like a camera, to allow light through. And then on an old microscope, it would be a mirror. But nowadays, most microscopes tend to have a light source that light can travel up towards your eye. Now, you may be asked to calculate the magnification. Well, it depends on what objective lens you are using. If I have a objective lens of 40 over here, then I've got to take into consideration the eyepiece objective lens and the objective lens. So 40 times by 10 means that my object is being magnified by 400 times. Really nice and easy. Just remember that the eyepiece has a magnification of 10. Now, there's some important terms they like to use, and it's so important that you can define these when it comes to the microscope, and that is magnification, the degree to which the size of an image is larger than it is in reality, and then resolution. Now, we know that there is the light microscope and there's the electron microscope. And because the electron microscope can see in so much more detail, the resolution is better. So resolution is defined as the degree to which it is possible to distinguish between two objects that are very close together. So in a light microscope, you may not see the mitochondria. But with an electron microscope, not only will you see the mitochondria, but you'll see the beautiful folds of the inner membrane as well, because it has a high resolution. Okay, now standard form from mathematics. You need to be able to make these conversions, because in biology we look at very small things that might be a nanometer big or even a picometer, 10 to the minus 12 of a meter. So I would memorize this. You know already that there are 1,000 millimeters in a meter. You would then have to times it by another 1,000 to get micrometers, which is this funny U sign here, micrometers, 10 to the minus 6. And then nanometers is another 1,000 times smaller, 10 to the minus 9. And then you get picometers, 10 to the minus 12. And by memorizing this, you can very easily do your conversions. And this is where they challenge you. If they give you it in one unit and expect you to give an answer in a different unit. So look out for that and don't get caught out. Right, now, will I am is always my light bulb moment to help me remember the equation for magnification. I am, will I am. So let's have a look at a question where I am using this particular triangle. It says here the diagram below is a drawing of an organelle. In fact, it is the mitochondria because of the folded inner membrane. An organelle from a ciliated cell as seen from an electron microscope. Calculate the actual length. So that's the A. What is the real, the actual length of the organelle as shown by the line AB in the diagram. Express your answer to the nearest micrometer. And actually, I should make that a UM. Apologies for that. Show your working. Okay, so let's just think about this first. If I'm calculating the actual size, from my triangle, I cover up the A, the I's at the top, so it's I divided by M, the image size divided by the magnification. So, using the ruler, and this is why you need a ruler and a calculator in your biology exam, you can measure the size of that mitochondria. And looking at it very, very carefully, it is 102 millimeters. Now, the problem is they've asked to express the answer in the micrometers. So we've got to convert millimeters into micrometers. If you remember, as we were going down, there are 1,000 micrometers in 1 millimeter. So we've got to times that by 1,000, giving us 102,000 micrometers. And then where do we get our magnification? If you have a look, it tells you that this has been magnified 200 times. So I put that in the calculator, and my answer is that this in reality is 5.1 micrometer. Let's have a look at one more example. The diagram below shows the general structure of an animal cell as seen under an electron microscope. Calculate the magnification this time. Okay, so remember, will I am, so M is I divided by A. What information do we have? We've got this little line here, and this is telling us that that line represents 5 micrometers. So the actual size is 5 micrometers. What is the image size? Well, that's when you need to take out your ruler. So what you will do is, using a ruler, that measures 24 millimeters. So I've got 24 millimeters and I've got 5 micrometers, my I over my A. Now, I need to make sure that they're both in the same unit. So I know there are 1,000 micrometers in a millimeter. So I times it by 1,000. Now they're both the same units. And folks, that is the most common mistake people make. Don't be silly like that. Okay, plug that into my calculator. And I'm always going to check this at the end of the exam in case I made a Wally mistake. And my answer comes to 4,800 times. This cell is 4,800 times bigger than in real life. That's how much it's been magnified. Now, I hope that is helpful. What I suggest, go through the summary, save it, and then practice questions. Well done. Keep smiling. And don't forget to subscribe to my channel for more of these revision videos.